Nearly 70% of all medical decisions depend entirely on laboratory test results. While a physician's physical examination forms a clinical hypothesis, the laboratory provides the quantifiable proof required to confirm it. Medical laboratory technology is the core analytical center of modern healthcare, providing the data that guides every stage of treatment. This profession manages a specific, high-stakes tension, converting raw, degrading physiological samples into precise data without any margin for error. To achieve a 0% error tolerance, a medical laboratory technician is systematically built through 3 rigorous pillars of development. The first pillar is a dense educational matrix. This tracks a student through both theoretical bioscience and intensive manual clinical training. The second pillar is an uncompromising code of ethics. Inside the laboratory, these protocols function as a strict clinical survival mechanism for the patient. The third pillar is the application of clinical biochemistry, the mechanical process used to translate physical matter into numerical data. This 3-pillar architecture ensures that modern medicine prevents fatal misdiagnoses and sustains the entire public health infrastructure. The foundational curriculum for a technician is a dual-track system, requiring intellectual rigor and physical precision in equal measure. Before students handle a live sample, they must master a map of theoretical biosciences, anatomy, clinical biochemistry, and pathology. They also study microbiology, the science of microscopic pathogens and tiny organisms, using tools that trace their lineage to Antoni van Leeuwenhoek's original 17th-century single-lens microscope. This theoretical knowledge is then translated into manual dexterity through mandatory on-the-job training, or OJT. Every trainee is measured against this rigid grading matrix using a 100-mark scoring system. It emphasizes cognitive abilities like problem-solving, balanced by strict requirements for manual skills and tool familiarity. During training, a failure in manual dexterity directly reflects a potential compromise of a patient sample in the field. The curriculum is designed to identify and remove physical or mental imprecision before a technician ever touches a live diagnostic sample. In an active laboratory, professional ethics serve as non-negotiable quality control mechanisms. The principle of absolute accuracy creates a zero-tolerance policy for guessing or making assumptions on a lab report. If a technician assumes a single variable, they provide flawed data that can lead an attending physician toward a fatal clinical misdirection. Confidentiality is a moral and legal protocol. Findings remain exclusively between the technician and the prescribing doctor to protect patient privacy and legal integrity. Principles of promptness and punctuality are vital, especially during emergency medical scenarios. In critical care, the speed of lab processing determines the patient's treatment window. A delayed test results in a delayed life-saving intervention. To protect the integrity of the results, the lab environment is strictly controlled. Technicians must wear protective gear, including white cotton aprons and gloves at all times. Smoking, eating, or drinking in the lab is strictly prohibited, as is allowing unauthorized visitors into the testing area. These rules prevent cross-contamination and hazardous accidents. This rigid code of conduct transforms a human operator into an operational failsafe, shielding the diagnostic pipeline from human error. The final pillar is applied clinical biochemistry, the science of quantifying biochemical constituents within body fluids. A technician's primary challenge is translating invisible chemical concentrations inside blood or urine into precise numerical data. They achieve this through photometric instrumentation, specifically a process called spectrophotometry. This relies on the Beer-Lambert law. Light absorbed by a solution is proportional to its chemical concentration. Introducing reagents triggers a color shift, altering light absorption, which the instrument measures with extreme precision. The resulting optical density value provides the raw mathematical data used to calculate the patient's exact physiological state. Spectrophotometry acts as an objective measuring tool, removing human bias and providing quantifiable mathematical proof of a disease state. These theories of clinical biochemistry are applied to specific high-stakes medical assays that dictate patient treatment. To analyze uric acid levels, technicians target 2,6,8-trihydroxypurine, the end product of purine metabolism. They utilize the Henry Caraway method to extract and measure these levels from a patient's blood serum. This quantitative data allows a physician to confidently differentiate between gout and non-gouty arthritis. Another critical assay is serum creatinine analysis, which is used to evaluate potential organ failure. Technicians use the alkaline pick rate method to find the precise creatinine concentration within the serum. This specific data point is the linchpin for confirming catastrophic conditions like renal failure or cardiac dysfunction. Mastering these specific chemical translations establishes the technician's diagnostic authority, enabling them to pinpoint microscopic failures before they cause systemic collapse. The medical laboratory technician is built upon 3 structural pillars: foundational curriculum, uncompromising ethics, and biochemical mechanics. These pillars work in unison. The curriculum dictates the test, the ethical discipline protects the sample, and biochemistry extracts the quantifiable facts. This rigorous pipeline results in a singular, flawless diagnostic report delivered directly to the physician. These professionals form the silent backbone of the healthcare system, executing the tests that support nearly all medical decisions. Their work directly prevents the spread of infectious diseases and ensures that complex treatment plans are based on accurate data. Although they operate entirely behind the scenes without direct patient contact, their analytical reports dictate whether an infection is identified or an organ failure is caught before it becomes irreversible. In modern medicine, the careful observation and flawless execution of the medical laboratory technician are the factors that ultimately save lives.